Next, it gives me great pleasure to introduce Dr. LaVoie. Dr. Ann Summers was unable to be here today. We are grateful, however, to have her associate, Dr. Stephen LaVoie, here to discuss their research. Dr. LaVoie earned his baccalaureate in microbiology from the University of Georgia, and he has worked as the project manager with Analytical Environmental Services in Atlanta. He currently works with Dr. Summers as a postdoctoral associate at UGA, researching bacterial proteins that are vulnerable to damage from organic and inorganic mercury. Dr. LaVoie has published several peer-reviewed articles, and he is here to share the research findings that he and Dr. Summers have completed. Please join me in welcoming Dr. Stephen LaVoie.
Hello, everyone. Thank you for the introduction, and sorry Ann couldn't be here, but I appreciate the opportunity to be here and present our research to you. They've asked me, I do not have any financial interest or disclaimers to make, so I think that should be covered. All right, so the title of my talk is Towards Biomarkers and Remedies for Mercury Exposure. Inorganic and organic mercury damages proteins differently, and cells respond differently as they recover from these toxicants. So what does that mean? Proteins do the work of the cell, and if proteins are damaged, then they are not able to function. Some protein structures, their structures make them more vulnerable to bind mercury than others, and so our aims are to identify the most vulnerable proteins in the cell, and to also assess if inorganic mercury and organic mercury cause different types of damage using a systems biology approach and looking for differences in how the cell recognizes these different toxicants. Before I get started, I first want to thank our collaborators. Almost all of these projects were done with collaborators in various institutions. Other members of our lab and Bob Scott and Mike Johnson in the UGA chemistry department helped with some of the biochemistry assays. Sue Miller and Ben Polacco helped with were part of the proteomics team and did most of the bioinformatics work. And Mary Lipton's group at Pacific Northwest National Lab for the proteomics analysis. There are other projects in the lab that are done in collaboration with the Oak Ridge National Lab and University of Missouri and Oregon Health Sciences, but I will not go into any details on those projects today. So first I'm going to take you through some background, then get into our findings, which look at what does mercury do to biological molecules, then how does the cell respond to mercury exposure, and finally some take-home messages. So first, for some background, what are mercury's preferred biological ligands? What are environmental sources of mercury exposure? And lastly, mercury detoxification in bacteria. So there are essential and toxic metals. Most of the biologically essential metals are shown in cyan, in the cyan box here. All of these will bind to the halides that are shown in orange, as well as to nitrogen and oxygen shown in red. As you move across the transition metal group towards, especially in group 12, shown here in blue, these bind most strongly to sulfur and selenium, so the amino acids cysteine and selenocysteine. Cadmium and mercury have no known biological function and are both considered toxic. So this is a picture depicting the global mercury biogeochemical cycle. This includes abiotic processes such as the oxidation of elemental mercury vapor in the atmosphere by ozone to ionic mercury, as well as biotic processes such as reduction, oxidation, methylation, and demethylation. And most of these processes are bacterial mediated and highlighted with the green arrows. The global atmospheric burden is roughly 2,000 tons per year with a 1.5 year half-life. Most of the anthropogenic sources of mercury emissions into the atmosphere comes from the burning of fossil fuels, which is almost 500 tons per year. and 25 of those tons coming from the US alone. There are a number of other industrial processes that contribute to mercury emissions as well as local contamination. All of these processes their emissions are regulated with the exception of artisanal gold mining and dental office amalgam waste in the US. So what's missing from this previous graphic is humans. Humans can be exposed to mercury through what they eat, where they work, and as well as from their amalgam fillings in their mouth. Approximately 30 tons of mercury is used in the US dentistry every year, and approximately 80% of the US population has at least one filling. The average number of amalgam fillings per person is 7, which is roughly 5 grams of mercury per person. So if you round the US population down to 300 million, multiply that by 80%, and then by 5 grams per person, that's roughly 1,200 tons worth of mercury in the mouths of US people, and that's not something that should be ignored. So the ground zero for mercury exposure in the body is the GI tract and the lungs. The kidneys and the liver load mercury most heavily, but almost all of the mercury that passes through the body passes through the GI tract and is excreted out. So this is important because the GI tract contains a robust and diverse microbial community that can convert mercury from its inorganic form to its elemental vapor form, as well as methylate the mercury, which allows it to cross membrane barriers and go into circulation. The GI tract also contains roughly 80% of the body's immune cells and serotonin receptors. So the GI tract, in a way, has a brain of its own and influences the body's well-being. And the more that we learn about how the microbiome affects the body, we're seeing more and more effects of maintaining a healthy microbiome in your gut. So it's important that the mercury that's passing through your GI tract not only can affect your own cells, but also affects the bacteria that live there. So how do the bacteria in the gut respond to amalgam fillings? We've previously shown that when amalgam fillings were installed in monkeys, upon this installation, we see spikes in the mercury concentration that's passing through the GI tract and being excreted out in the feces. And this concentration spikes to millimolar levels and diminishes over a 5-week period until the amalgam fillings are removed at 8 weeks. And then we see a secondary spike for a secondary exposure. And this corresponds to a delayed response, but an increase in mercury-resistant bacteria within the gut. So as the microbe population turns over in the gut, we see an increasing abundance of mercury-resistant bacteria, and until these fillings are removed, and therefore losing their competitive advantage, and the selective pressure diminishes once the amalgam fillings have been removed, and these mercury-resistant bacteria go back to their normal levels within the population. So, metals homeostasis and metals resistance is in all other systems as a defense mechanism that relies on proteins that can efflux or pump out metals from the cell. Mercury resistance is very different from that. The mercury resistance system, or operon, encodes genes that encode transporters that will actively bring mercury into the cell, where it can be reduced from inorganic mercury to elemental mercury Hg0, and which is relatively inert and will diffuse out of the membrane. And these genes are regulated, the transcription of these genes are regulated by MER-R and MER-D. So these genes will only be expressed when mercury is present in the cell. And as soon as MER-R binds very tightly to mercury, and so as soon as any mercury enters into the cell, it turns on transcription of these genes, which allows the cell to detoxify its local growth environment. And so this is very different from all other metals, where the cell can only just keep it out, but can't actually do anything with it to get rid of it. This mercury resistance set of genes is common throughout the entire prokaryotic branch of the tree of life. You can most easily see these here by the blue, which represents the MER-A gene that's responsible for reducing the mercury. The most complex and advanced merloci are found in the proteobacteria, and more ancient bacteria have simpler systems with fewer genes. The mercury resistance is notably absent from anaerobic bacteria and from phototrophic bacteria because their local environments are either high in sulfur or can rely on photoreduction to remove mercury from those environments. And most importantly, this mercury resistance is absent from eukarya, which includes plants, animals, and most importantly, humans. And so this is not a defense mechanism that's available to us. So far what we've learned is that mercury binds to sulfur and selenium. The environmental sources of mercury exposure are natural, industrial, and from amalgam fillings. And lastly, that mercury resistance is only found in bacteria and not available to humans. So next, what does mercury do to biological molecules? What does it bind to, and what effects does this have on cellular physiology? So the primary targets of mercury, once it enters a cell, is to bind to proteins. This cartoon here shows a cross-section of an E. coli cell drawn to scale. These are done by David Goodsell, and he has a number of really great illustrations of all sorts of biological systems. So mercury can bind to outer and inner membrane proteins, as well as ribosomal and cytosolic proteins, and components of the nucleoid, which includes DNA, RNA, and proteins. So the mercury compounds that I've used in this study and we'll talk about and we'll mention throughout the talk are mercuric chloride, which is our inorganic mercury compound. So this is inorganic because it is a mercury bound to 2 chloride atoms, and these chlorides will be exchanged for other ligands such as sulfurs. The organic mercury compounds are phenylmercury, or PMA, and ethylmercury thiosalicylate. This is more commonly referred to as methylate or thimerosal. These are organic mercury compounds because of the mercury carbon bond that's found in each of these. So this mercury is bound to a phenyl group, and this mercury and methylate is bound to an ethyl group. And these will not be exchanged once it enters the cell. So how are organic mercury, noted here as RHG, and inorganic mercury, noted as Hg2, toxic? They're toxic due to their high affinity for sulfur and selenium ligands. So here I'm showing affinity constants, and these are on log scale, so bases of 10. And the larger the number, the stronger the bond that the mercury makes to these ligands. So the weakest ligands are to nitrogen and to oxygen for acetate here, and mercury increasingly binds stronger to halides as they increase in atomic mass from chloride to bromide to iodide. And so looking at thiols are sulfur ligands such as the amino acid cysteine or the tripeptide glutathione. We see that this number is much higher. This 35 to 40 range is, because of this is on log scale, is over 1 trillion times stronger than this highest affinity constant for iodide. So that's why these weaker ligands are exchanged and mercury will bind to the sulfur ligands that are in the cell. Mercury also binds very strongly to selenium, such as the amino acid selenocysteine, and mercury is bound also very strongly when it is chelated by 2 thiols, such as in a protein where 2 cysteine residues are in close proximity to one another and can chelate this and stabilize the mercury. And so this bond is very strong, and this is also found in a number of synthetic chelators that have been developed, such as DMPS, DMSA, and NBMI, which I believe that Boyd Haley will be discussing later on this afternoon. So even though mercury binds very strongly to these monothiols, mercury can be exchanged between them very rapidly. So mercury that's bound by, say, 2 glutathiones here in the presence of a third can form this transient trigonal complex, and one of these thiols is exchanged for the other. And this happens rapidly within the cell. But when mercury is chelated, shown here, this blue squiggle represents a protein. And when there are 2 cysteines that are in close proximity, they can chelate this mercury. And this mercury is very hard to remove. If another glutathione or cysteine comes in proximity to this. Mercury is bound very stably and not going to go anywhere. Cysteines play important roles in most of the proteins that you find them in, and so this mercury being chelated between there will likely inhibit the protein's ability to function. So this is our experimental setup for our biochemical assays and for our proteomics. I'm growing mercury-sensitive E. coli cells in liquid medium and then exposing them to various concentrations of inorganic mercury or organic mercury for 15 minutes, and then harvesting these cells and running a number of assays on them to see what the bulk effects of these different mercurial exposures are. This includes total thiols, protein thiols, essential metals content, and the free iron content of the cell. I will discuss the proteomics effects later on in the talk. So how do we see protein thiols? Here I've drawn a cartoon to illustrate this, where the blue represents a protein, the orange triangles are cysteine residues, and we use a fluorescent dye that will bind specifically to thiol groups, such as cysteine. So here, with the addition of this fluorescent dye, the dye will bind to the surface exposed cysteine residue, but if there are interior cysteine residues that are buried within the protein that might not be solvent accessible and will not be bound by the dye and not be visible to detection by the and emit fluorescence. So in order to see all the cysteines within this protein, we can use urea, which will denature or unfold the protein, and this will allow us. And so this protein would have twice as intense, or the intensity of the fluorescence would be twice what the one above it would be. So how does this look when mercury is added? Mercury will bind to these cysteine residues, and when we add the fluorescent dye, it will not be able to bind because that site will be blocked by the mercury. And so therefore this protein here would emit no signal whatsoever. And again, we can use urea to denature, and we might get some increase in fluorescence for any buried cysteines that were unavailable to the mercury during growth. So what does this look like in practice? Here I'm showing that inorganic mercury blocks thiols much more effectively than PMA and methylate. So focusing on lane 1 first, this is proteins from a cell that was not exposed to any mercury at all, and with no urea used. The proteins are separated out on a gel where the largest proteins are at the top and the smallest are at the bottom. And so these are all the thiols that we're able to detect from various proteins within the cell. And the dark bands are the fluorescent signal. When we add the urea to unfold the proteins, we see additional bands show up and the intensities increase. So with the addition of the mercuric chloride exposure during growth, we see almost all of this fluorescence intensity diminish, indicating that most of the cysteines on the proteins were blocked. And it requires twice as high a concentration of PMA to achieve the same level of blockage. And the intensity never gets anywhere close to as diminished, even at 160 micromolar for methylate, so it's much less effective at getting into the cell and binding to these protein thiols. So, and a number of essential proteins also coordinate metal cofactors, iron being one of the most common. These proteins that bind iron serve a number of essential roles in the cell, from respiration, energy production, and for DNA repair. So we wanted to look at what the effects of mercury are on these metal cofactors. So in this chart up above, I'm showing the fold change and increase in free iron as measured by EPR relative to the unexposed condition. So we see that with the addition of, for 4 millimolar hydrogen peroxide exposure, which is a common oxidizing agent that will induce oxidative stress, we see a fairly minimal increase in the free iron in the cell. And the iron in the cell is typically bound because if it's bound by proteins, because if it's in its free state, it can undergo Fenton chemistry and catalyze the formation of superoxide, hydroxyl radicals, and this will do additional damage to proteins, DNA, and RNA as well, or in lipids. So as well with tert-butyl hydroperoxide, which is also an oxidizing agent, we see minimal increases in free iron. And with 160 micromolar methylate, we see insignificant increases in free iron. So this reflects what we saw on the previous slide, where methylate is not very effective at binding to these protein thiols. But at 40 micromolar PMA exposure, we get nearly a twofold increase in free iron. This is 100-fold less concentration than the 4 millimolar hydrogen peroxide, so it's much more effective than these common oxidizing agents at disrupting iron from proteins. And as little as 16 micromolar mercuric chloride results in an over twofold increase in free iron, and 80 micromolar mercuric chloride exposure results in a greater than threefold increase in free iron. So this would be roughly 80% of the iron pool in the cell would be displaced from proteins. So here I have a cartoon to illustrate what we think is happening here. So the green represents a protein structure. These arms here, there are 4 cysteines, 1, 2, and 3 here, and there's a 4th and behind this. They coordinate this iron-sulfur cluster. The irons are in red, and elemental sulfur is in yellow. And so these are essential for doing redox reactions and electron transfer reactions within the cell. And with the addition of mercury, this iron is displaced from the cysteine, and the mercury binds to it and releases the iron from the protein. And it's also possible for the entire cluster to be displaced, and additional mercury could either bind to these other cysteine sites, or if the cysteine residue is in a proximity, it can chelate this mercury right here, and therefore that would prevent the the protein from being repaired and that cluster from being reinserted. So the protein would no longer be functional. So here are some examples of proteins within the cell that bind iron normally, but that we observed with mercury bound and the sites that coordinate the iron. This is from our proteomics work that, as I mentioned, I will discuss in more detail later. So here we see that there are a number of proteins from 6 different functional categories that normally bind iron. Most of these are from energy production and amino acid metabolism. And what's most important to note here is that everywhere that we see inorganic mercury adducts, it's where there are 2 cysteines in close proximity that can chelate that mercury, whereas the organomercurials make single cysteine ligands that can be found throughout the protein structure. So we see that, so this confirms what we believed before, where the iron is being displaced, and we can see with the proteomics that mercury is actually binding to these sites that coordinate the iron and damage the proteins. We also looked at the total metals content of the cells, where we saw that 6 essential metals showed no significant changes in their bulk metal content. So even though we see that iron is being displaced from proteins, it's not being lost from the cell in any significant amount. It's still in there, where it can potentially cause further oxidative damage. We did see significant changes in potassium and sodium. So looking at the inset up here, we see that under mercury exposure conditions, we see a 50% decrease in potassium and a threefold increase in sodium. Potassium and sodium are important for maintaining the electrolyte balance of the cell and therefore pH. So at least in E. coli, we know that there is a potassium transporter, KEF-C, or a potassium antiporter, KEF-C, that when the cell is under oxidizing conditions, it's regulated by glutathione. The glutathione is oxidized or bound by some electrophilic chemical, and this induces the antiporter to pump potassium out of the cell and bring protons in, which will decrease the intracellular pH of the cell to protect it as a defense mechanism. And we think that the increase in the sodium is the sodium antiporter in HAA trying to maintain the pH balance by bringing in sodium ions and pumping out protons, which will normalize the intracellular pH. So we saw that mercury can bind to protein thiols and displace good metals, and we also see that because of the mercury's ability to displace these good metals from proteins, that it can amplify a single mercury atom's damage by inducing reactive oxygen species formation. So next, we'll look at the proteomics. This allows us to see all the proteins in the cell and identify individual proteins that were bound by mercury. So how does this work? Seeing all the proteomics allows us to see all the proteins in the cell at once. To do this, we have proteins, they're shown here. They are digested by trypsin into peptide fragments and then analyzed by liquid chromatography tandem mass spec, and we will get a spectrum here, and this spectrum is matched to the theoretical spectrum that is derived from the protein sequences that's derived from the genome sequence of the organism. We can match these together and identify which peptides belong to which proteins. So with the addition of mercury, where there is mercury bound to the protein and this peptide fragment, we will see a shift in the spectrum by 200 Daltons due to the additional mass of the mercury being bound to that peptide. As well as that mass shift, mercury has 7 stable isotopes. So most other elements, hydrogen, carbon, nitrogen, oxygen, and sulfur, have 1 highly abundant isotope, but mercury has 7. So what this looks like in proteomics, or in the mass spec, is that a unmodified peptide will have a spectrum like this, and when mercury is bound to that same peptide, we'll see a shift in the mass due to the extra mass of the mercury, and we also see a change in the isotopic distribution. So our collaborator, Bim Polacco at UCSF, developed a computational filter that allows us to identify these mercury adducts within the proteomics data. So our experimental setup is the same as before. I'm growing mercury-sensitive E. coli cells, exposing them to either 20 micromolar mercuric chloride or 40 micromolar PMA, or nothing at all. For 15 minutes, cells are harvested and then prepared for proteomics analysis. We are operationally defining a stable mercury protein adduct as one that we can detect by mass spec. So there may be additional proteins that are bound by mercury, but we can't observe them if they can't make it through the process and through the mass spectrometer. So this table summarizes our proteomics observations. E. coli encodes roughly 4,321 proteins. We observe 1,556 of these. This is better than the work that's been, this is better than what has been observed by others' work. And we don't expect all of the proteins to be expressed all the time. What proteins are being expressed depend on the growth conditions. Of these 1,556 proteins, 641 of them had a cysteine residue. And of these 641, 307 we observed with mercury modifications on them. So that's roughly half of all proteins that encoded a cysteine that we were able to observe was modified by mercury. So this greatly exceeds what's been observed in the past by others. Typically, only 10 to 15 proteins have been observed by other methods that have been modified by mercury, and here we have a data set of over 300 proteins that were modified by mercury. So what's the distribution of these proteins in the cell, and what are their functions? So here on the x-axis, I have functional categories. These can be grouped into the hierarchical categories of information storage and processing, cellular processes and signaling, metabolism and transport, and even though E. coli is probably the most well-studied organism biochemically, there are still a number of proteins that have been not well defined yet. On the y-axis, this is the percent of the indicated group total. So for all the proteins encoded in the genome, shown in blue, we see that 5% of the genome encodes proteins that are involved in translation, and around 6% are involved in transcription, and so on. The green bars are the proteins that we observed, and so we see the distributions of these are different, where we see that almost 11% of the proteins we observed are involved in translation, translation proteins being more abundant compared to some of these other categories that may or may not be expressed. And the red bars are mercury-modified proteins. So we see that 19 of the 23 functional categories were observed with mercury modifications. So there are not specific functions that are vulnerable to mercury. It is typically the cysteine-rich categories that tend to be rich in mercury adducts. But all functional classes and proteins are vulnerable to mercury damage. So some specific examples of proteins in each of these categories are, in translation, 14 ribosomal proteins, 10 tRNA synthetases, elongation factors, and initiation factors. So without the ribosome and these other proteins, the cell will not be able to make new protein. In transcription, we observe 3 RNA polymerase subunits bound by mercury, sigma factors, termination factors, and RNase 2, which is involved in message turnover. So for these, transcription will be inhibited. If these are damaged by mercury, then transcription will be inhibited. The cell will not be able to make new messages and turn those messages over properly, and will interfere with a cell's ability to recover. Energy production was the second most abundant category, where we saw 3 ATP synthase subunits, as well as a number of enzymes involved in the TCA cycle that were modified by mercury. So if these pathways are disrupted, then the cell will be starved for energy, and that will also inhibit its ability to recover. And lastly, the most abundant category was amino acids, where we saw 8 different amino acid biosynthesis pathways. Some of these amino acids, there were multiple proteins that were damaged by mercury, and so this also interferes with the cell's ability to make new proteins as well. So out of those 307 proteins that we saw with mercury modifications, are any of these found in humans? The answer to that is yes. Here's a subset of some of them that are of E. coli proteins that are also found in humans. The 11 shown here have a greater than 50% homology to the peptide from the E. coli protein to the human protein, and these all have diseases associated with mercury exposure based on the comparative toxicogenomic database. In total, we saw 33 sites from 25 human proteins that could serve as potential biomarkers for mercury exposure and other higher organisms, and 13 of these that are shown here in bold with the red M are from mitochondria. So using proteomics, we were able to find a number of the most vulnerable proteins to the cell to mercury that could serve as potential biomarkers for exposure. Next, we wanted to look at how does the cell respond to mercury exposure transcriptionally, and to see whether the cell is able to recognize inorganic mercury and organic mercury differently in its transcriptional response. So for this, here's our experimental design. I'm growing, again, mercury-sensitive E. coli cells in liquid medium to the middle of their growth phase, and then exposing them, this time to very low level or subacute concentrations of mercury at 3 micromolar mercury chloride or at 3 micromolar phenylmercury. At 0, and after this exposure, I'm collecting samples at 0, 10, 30, and 60 minutes, and quickly storing these cells and then preparing them for RNA sequencing analysis, where we will do differential expression analysis, which I will explain on the next slide. So this is a growth curve. So along the x-axis, we see time, and on the y is turbidity or optical density. So we see from 0 to 200 minutes, the culture is growing normally. At this point, the mercury was introduced to 2 of the cultures. The one that had no mercury is the blue line and continues normal growth, and the PMA and the mercury exposed cultures show growth being inhibited. So we operationally define a subacute concentration of mercury here as one that is high enough to inhibit the growth rate, but low enough to allow the cells to recover growth within 1 hour. So just focusing in on this exposure period here on the next slide. So here it's the same thing, it's just on the time axis, 0 is where the mercury was first introduced. The blue again is with no mercury added. And we see that for the PMA exposed culture, growth is inhibited at 10 minutes, but the cells begin to recover growth within 30 minutes, and by 60 minutes the growth rate matches that of the unexposed culture. For the inorganic mercury exposure, growth is inhibited much longer. We don't begin to see any increase in growth rate again until 45 minutes, and it takes until beyond 75 minutes for the growth rate to actually recover back to normal. So on the next slide, I have some So for differential expression analysis, what I'm doing is comparing expression of all the genes under the mercury exposure condition relative to the unexposed condition at each time point. So gene expression here, compared to here, and again for the subsequent time points. So on this next slide, I have some Venn diagrams that show a summary of the differential gene expression. On the left column is upregulated genes, and on the right is downregulated at 10 minutes, 30 minutes, and 60 minutes after exposure. The red circles are the inorganic exposure, and the blue are PMA. So we see for the inorganic exposure, there were roughly 900 genes that were upregulated at 10 minutes in response to mercury, and over 1,200 genes in response to PMA. And over the first 30 minutes, there were more genes that were unique to the PMA exposure than shared. So both here and here, these numbers are greater than the shared value. But there were more shared for the mercury than there were unique. By 60 minutes, where growth of the PMA culture was back to normal, there are very few differentially expressed genes, but there are still almost 250 genes that are being differentially expressed after an hour in response to inorganic exposure. For the downregulated response, is much more shared for both compounds. There are more genes that are shared than unique for both over all time periods. And again, we see very few differentially expressed genes for PMA at the 60-minute time point, whereas there's still 288 for the inorganic exposure. So we see that there is some overlap in their transcriptional responses and how the cells recognize these two different toxicants, but there are also distinct unique factors for both of them. So this image shows a heat map of all differentially expressed genes for each condition. So along the bottom, I have inorganic exposure at 10, 30, and 60 minutes, and PMA exposure at 10, 30, and 60. The legend here on the far right can be thought of like a thermometer, where the black is no change in expression from the unexposed, and as you increase expression, the brightness towards yellow shows a greater increase in expression, and from blue to cyan, a greater decrease in expression relative to the unexposed. And these are log base 2, so a 10-fold change would be roughly a log base 2 fold change of 10 is equal to a 1,000-fold change in expression. So looking first at the PMA exposure, because it's simpler, there is a large block of upregulated genes and a large block of downregulated genes that are largely shared over that first 30 minutes. And by 60 minutes, there are very, very few differentially expressed genes compared to the unexposed culture, because the growth rate has gotten back to normal. Looking at inorganic mercury exposure, we see some of these genes are shared with PMA, but the response is much more variegated over time. We see that there are genes that are unique to only the 10-minute time point, and 30 minutes and 60 minutes as well. And so the cell is struggling much more to adapt to this type of exposure and having to alter its transcriptional response over time much more to recover. So here I have a subset of some of the differentially expressed genes grouped by functional categories. There were a lot of these, but these are just some highlights. The heat map on the left is largely downregulated functions, and the heat map on the right are largely upregulated functions, and the legend is the same from the previous. So here we see that ribosomal proteins, nearly all of the ribosomal proteins were downregulated in response to mercury, whereas only some of them were for PMA. Motility, nearly all the genes were downregulated for motility for both compounds, but the intensities were much greater for PMA. Electron transport and energy production, as well as amino acid biosynthesis, metabolism, and metals uptake were all largely downregulated, but there are some distinct differences between the two compounds. We see here in amino acid biosynthesis that methionine and histidine biosynthesis genes were upregulated in response to inorganic mercury, but downregulated in response to PMA. And the opposite was true for leucine and isoleucine biosynthesis. We don't have clear explanations for this yet, but there are differences in how the cell responds transcriptionally to these different compounds. Looking at upregulated functions, we see heat shock, all of the heat shock proteins and chaperonins being upregulated in response to inorganic exposure. These heat shock proteins and chaperones are responsible for repairing damaged proteins and protein turnover. Metals transport, as an efflux, is largely up for both compounds. And iron storage, because we saw earlier that mercury can displace iron, we see the iron storage proteins and iron sulfur cluster assembly and repair. All of these genes are up for the inorganic mercury because it was more effective at damaging these proteins, and only one of the repair systems is upregulated for PMA. Redox homeostasis is largely up for both compounds, but PMA also had some more distinct effects on multi-drug resistance. So these are proteins that efflux various antibiotics out of the cell as protective mechanisms, and also to surface structure such as biofilms and fimbriae, which are involved in cell attachment. There were a lot more of these genes that were upregulated in response to PMA than to inorganic mercury. So it doesn't matter what form of mercury is poisoning you. Based on our biochemical assays, we see that on a molar basis that inorganic mercury is more effective at damaging proteins than PMA or methylate, as based on their thiols and their iron homeostasis and electrolyte balance. And large excesses of mercury in the cell, we have observed that mercury can bind, when thiol ligands become depleted, that mercury can bind to nitrogen compounds as well, such as DNA, which it did not show. So, yes. As for the proteomics, PMA and methylate made stable adducts to many single cysteine peptides, but inorganic mercury was only stable to be identified when chelated between two cysteines. Mercury and PMA both formed formed adducts with distinct proteins in nearly all the functional categories, but notably energy production, amino acid biosynthesis, and redox defense. So yes, here again. And lastly, transcriptomics. The cell's strongest responses were qualitatively distinct between the equal molar concentration exposures of inorganic to PMA, and many of these differences were quantitatively larger for inorganic mercury than for PMA. The largest transcriptional responses occurred in genes that were found to be vulnerable to mercury in the proteomics analysis, and the cells recover more rapidly from the organic mercury exposure than from the inorganic. So yes, here again. So what's in the works for the future? There's a project proposal for studying epigenetics effects of inorganic and organic compounds in eukaryotes. This would be using fungal and mammalian tissue and organ culture models, looking at mercury-vulnerable peptides and histone methyltransferases. And this will allow us to learn or identify which potential eukaryotic peptide markers, or identify potential eukaryotic peptide biomarkers of real-time mercury exposure. And a grad student in the lab is following up on the transcriptomics work, looking at how transcription changes in mercury-resistant cells. So this will show us how the presence of the mercury locus will affect RNA expression during recovery, and which chromosomal pathways and genes does the MER enzyme spare. So this will help us to identify metabolites required by the cell during recovery and to guide future work of nutritional and pharmaceutical support for higher organisms which lack the MER locus. So I rushed through that a little bit, so we're done a little early. So there's plenty of time for questions if anybody has any. But thank you for your time and your interest.